Great. And once again, thanks everyone for coming out. Welcome to our second Amplifying Voices webinar. So the Amplifying Voices webinar is a new initiative started by ASLO and specifically the Early Career Committee. My name is Kale. I'm one of the Early Career Committee members, as well as a few more members who are on the call today. Through our work and the support of the ASLO board, we've started this webinar series to highlight and specifically amplify the voices of early career aquatic scientists from across the world, specifically those from historically excluded groups, including to ethnicity or indigeneity, ability, sex, gender, or orientation, and any other sort of historically excluded identity or group. We had our inaugural session last month to a great success. So if you were here last time, or if you heard about that and watched it on YouTube after, we thank you very much for doing that. And we're very excited today to have two more great talks from early career researchers from India. Before I introduce them and begin, I just want to highlight that with the support that we've received, we're going to continue doing the Amplifying Voices webinar into 2022. We have scheduled talks at the end of January and the end of February, as you can see on this slide. And thank you all for attending. You know how to register, but if you are an early career researcher who is interested in applying to be a speaker, please contact us, the Early Career Committee, at our email listed on this slide and also this one. Because next month, we're going to hear from Justina Hampel from Sweden about some microbial communities in marine environments. But our second slot is still open. So if anyone, if you have research that you're interested in presenting to the ASLO community, then please do so. We would love to hear from you and get your research out into a wider audience. But today we're going to hear from two speakers from India. The first will be Dr. Aasif Mohammed Lone. Dr. Lone got his bachelor's in geological sciences from University of Kashmir before moving on to an MSc in applied geology, also at the University of Kashmir, and then doing a PhD also in geology at Anna University, Chennai, all in India. He now is a postdoc from IISER in Bhopal, India. He's going to talk to us today about Holocene paleo environments in the Himalayas, and I'm very excited for that. And then our second talk will be from Dr. Bhakti Salgaonkar, who is going to be presenting her research about Halophilic Archaea and some interesting things regarding bioplastics. So these extreme organisms will be very interesting to see. She got her undergraduate degree in microbiology from St. Xavier's College, affiliated with Goa University. Also at Goa University, she got a postgraduate degree in microbiology and then a PhD in biology from the Birla Institute of Technology and Science at Pilani University. She postdoced at the same institute, and then now is an assistant professor in the Department of Microbiology at Goa University in Goa, India. So I'm going to stop sharing my screen now. We're going to have Aasif, if you could share yours, and then we'll begin with his presentation. As he is getting ready, how this is going to work is we'll have about 20 minutes presentations. So Aasif will present. We'll have a brief 5 to 10 minute question period. Then Bhakti will give her presentation, another brief period of questions, and then at the end, we'll take some time to do an overall discussion about what we've learned and any questions we have, and it'll be a great time. So with that, I'll turn this over to Aasif. And if you have any questions, please put them in the chat or save them until the end of the presentation.
Hi everyone and good evening to all. My name is Aasif and I am an early career researcher. First of all, I would like to thank the organizers for having me and giving me the opportunity to share my research reports on aquatic sciences with the ASLO community. So for the next 20 minutes, I will be guiding you through my presentation, the title of which is the late Holocene lacustrine proxy records from Northwest Himalayas and their implications to reconstruct the regional hydroclimate variability. So first of all, I'll start with lake sediments, importance of lake sediments as paleoclimatic archives. So there are as we know, there are a number of controlling factors and processes that take place within a lacustrine basin. These controlling factors can be geology, climate, and the human impacts, and the process can be through the atmospheric deposition, the fluvial or the detrital deposition, as well as the slumping or turbidite or resuspension within the lacustrine basin. So together with all these, the lake deposits have been increasingly used in recent years to infer past fluctuations in climate, and all these previous studies demonstrate the great value of lake sediments as paleoclimatic archives. However, the response of lake sediments to climate is dominantly modulated by hydrological and geomorphic settings and by catchment geology. The transport of materials from source to the sink areas is dependent on climatically controlled hydrology. Once these sediments are deposited in the sink areas, they provide information or they incorporate information as proxies relating to the sedimentary structure and the composition. So see, this photograph shows uh, the lake sediment coring and the analysis. Once the cores are collected, they can be analyzed for a multitude of analytical processes and measurements. Once these sediments are deposited in as annually deposited sediments in the lacustrine basins, they can provide clues and answers to a variety of limnological questions, such as what is the range of the natural climate variability in terms of the spatio-temporal scale, what were the pre-disturbance conditions, that's the background conditions, have there been um, any changes in the paleo-environmental conditions? If yes, and how fast and how much? Is there any evidence of the human activity? How much improvement can be detected using modeling future conditions? And whether there has been any regime shift, such as the abrupt or non-reversible changes in ecological conditions that can lead to eutrophication or lake desiccation. So before answering those questions, we need to know what are the paleolimnological approaches that need to be undertaken. First of all, we need to study select a study site where we can do the coring or retrieve the sediments. Following this, we need to date the sediment or section the sediments and date the sediments using a variety of dating techniques. Then we can also do the sediments, do the sediment measurement uh, analytical techniques, and the data can be analyzed as a proxy records. This image shows the map of the Himalayas together with the Hindu Kush Himalaya. The Himalaya is literally termed as the abode of snow, and it spans across 8 Asian countries. This is actually a 2,400 long stretch that that spans across 8 Asian countries. It is very important in terms of it is a major contributor of Asian monsoon and cryosphere meltwater to a number of Asian countries. The climate also changes on on smaller distances. The climate is tropical or subtropical in the foothills to permanent ice covered peaks at the higher altitudes. As a result of this the climate has been defined as the climate has been defined as microclimatic zones in the Western Himalayas. Significant elevation changes also occur with distance. As a result of this, there is different rate of change of the temperature increase in different topographic areas. So this is the image that has been taken from the paper by Demiri and Dash, where they have in 2012, where they have uh, divided the Himalayas into three important zones. That's the Upper Himalaya, Middle Himalaya, and the Lower Himalayas. The Lower Himalaya is the area which is directly under the influence of the mid latitude westerlies. That's they get the uh, high amount of this snowfall or the rainfall through the mid latitude westerlies. Then we have the middle Himalayas and the upper Himalayas that receive significantly lesser rainfall compared to the lower Himalayas. So the Himalayas are also regarded as the best big reservoirs of the surface water, of surface fresh water, actually the outside the poles, in terms of the proglacial mountain and the valley lake systems. There have been a number of lakes. As you can see in this picture, these dark colored spots show the water basins within the Himalayas. A previous study, however, it is assumed that there has been a lot of change, paleoclimatic or paleoenvironmental changes occurring within the Himalayas, that is having a negative impact on these water resources. For example, a recent study by Abdullah et al. In 2020 suggests that there has been widespread changes in the glacial thickness during the period for which they studied these glaciers from 2000 to 2002 in the Western Himalayas. From their study, it is concluded that out of the 12,000 glaciers having around 19,000 square kilometer area have thinned, and they suggest that topography plays an important role in the glacier thickness changes in the Western Himalayas. Not only this, it has also these changes in the environmental conditions or the uh, temperature conditions have also led to changes in the high altitude lake hydrological conditions, that is the lake levels. A recent study by Srivastava et al. In 2020 suggests that out of the 13 lake sites that they have studied, uh, it is observed that 10 of the lakes show an increasing trend, and whereas 3 lakes show a decreasing trends in the water levels. This increasing trend was a result of the glacial melt or runoff due to high mean seasonal or annual temperature in the Western Himalayas. So when we look at the recent monsoon dynamics, that's the temperature and the precipitation trend variability in the Himalayas, it is seen that the Himalayas are significantly warming at a higher rate compared to the central Indian landmass. Not only this, the precipitation also shows significant positive and negative trends during the recent past. As you can see in this picture, this shows the temperature anomaly for the Indian landmass as well as the Himalayas. The Himalaya shows a higher rate of change in the temperature compared to the Indian landmass. This study was taken by Yadav et al. In 2011 and published in Climate Dynamics suggests that there has been widespread severe droughts and pluvial conditions between 15th and 16th century and the 17th and 18th century respectively. That is, this study is based on the tree ring proxy records from the Western Himalayas. These studies suggest that unfavorable conditions for the glacier with less accumulation and fast. This study actually suggests that these conditions have led to unfavorable conditions for the glaciers with less accumulation and faster melting. As a result of this, the equilibrium line of altitude have also moved up in the Western Himalayas. This could result in potentially large reduction in accumulation area as well as the small storage buffer of the permanent ice and snow in the Western Himalayan region. So as a result of this, there has been a lot of hue and cry as far as the Himalayas are concerned. because as a result of this increase in temperature and the drought areas, we are seeing that the warmer winters in the Himalayas are triggering avalanche. A recent study from the Snow Avalanche organization in India have suggested that winter temperature in the northwestern Himalayas have risen on an average of 0.65 degrees Celsius over a period of 25 years from 1991, which is significantly higher than the global average of 0.44 degrees Celsius. This rising temperature has led to an increase in the frequency of the avalanche since 1970, posing a threat to the regional infrastructure. Not only this, there has been also a significant amount of glacial retreat and the loss in the glacial areas during the 1960s to 2000 in different regions of the Himalayas. This retreat ranges from around few meters to almost 61 meters per year. The record suggests that a loss in area in the Western Himalayas has been about in different regions is around 13% during the last 4 decades. Now, as far as the climate history is concerned, the analysis of the trading proxy records also suggests a decline in the summer monsoon for the last 180 years in the Himalayas. For example, these two studies, that was one is from the Yadav et al. Paper and another one by Bhattacharya and Yadav in 1999. They are from the both of these studies used the tree studies from the Western Himalayas suggest that there has been widespread positive and negative shift in the temperature and the precipitation anomaly in the Western Himalayas. Not only this, Malik and Sukumar in 2021 could reconstruct the mean July temperature from Srinagar, which is located in the Western Himalayas, and suggest that they could pinpoint 10 cooler and warmer years during the period of 1773 to 2012 CE. So, another study by Dhyani et al. 2021 suggests that there has been increased or slow temperature relating to higher and lower growth rate during the medieval warm period, little ice age, and the recent warming during the last decades, which is also regarded as the recent warming trend. Okay, so how do we look back at the paleo environmental conditions beyond this time frame? We know that instrumental records, they span only for the last 300 years. The trading proxy records, they span only for the last 1000 years. We need long-term records to study the paleoclimatic variability. For that, we need the proxy record that can go beyond in time and space for a longer time period. So for that, we use the lake sedimentary proxy records from the Himalayan region. So this map shows the lake sediment studies that have been carried out from the Indian landmass, as well as some of the studies that have been carried out in the Himalayan regions. So this paper by Mishra et al. Actually, they compiled the lake sedimentary records of the using 76 lake sites from the Indian landmass, as well as from the Himalayan region. They could see that there has been optimum climatic condition, that the strengthening of the monsoon conditions from last 13,000 years to almost up to 4,000 years BP. But after that, there has been an increasing trend towards the arid conditions in these lakes lake sites. Another study by Kumar et al. In 2020 suggests that there has been during the last 11,000 years, around 21% of the precipitation in the Himalayas was dominated by the Indian summer monsoon, whereas around 80% of the precipitation out of the total precipitation was dominated by the mid-latitude westerlies in this region. Taking a cue from these studies, we are looking for the we try to look for the hydroclimatic and paleolimnological records from the Northwest Himalayas using lake sedimentary clues. So we have been collecting lake sediment records, lake sediment sequences from the Kashmir Valley, which is located in the northwestern Himalayas. This image shows the map of the Kashmir Valley, where we have the variety of lake systems, such as these are the valley lakes. Actually, we have the Manastal Lake, the Dal Lake, the Anchar Lake, and the Sampro Glacial Gangbal. So what are the possible sample collections we use to we take samples from the open lake systems. This is the Manastal Lake. The lake margin trench sampling was also done, as well as the Paleo Lake bed exposures were also we collected sediments from Paleo Lake bed exposure also to reconstruct the late Holocene paleoclimatic history. So this image shows the lithologue of the Anchar Lake, which was collected from of the sediment core, which was collected from the Anchar Lake. It is a 1.4 meter core and was dated to last 6,000 years. And this image shows the multi-proxy records that were derived from this sediment core. We analyzed the sediments for organic matter, calcium carbonate, C-band ratio, and some biogeochemical proxies. Based on these multi-proxy records, we say there has been three important phases that record increase in the monsoonal conditions with wet climate and gradual strengthening of the westerlies corresponding to the mid Holocene warming. That is the first phase. Then during the second phase, again the similar conditions were were seen, although a less uh, strengthening of the lesser strengthening of the monsoon and westerlies was observed during this time period. But during the latest of the late Holocene phase, that's the last 500 years, we could see that there has been increased terrestrial or anthropogenic input in this lake. As a result of this, there has been widespread desiccation and expansion of the meadows during this phase. Combining these multi-proxy records, we could reconstruct the paleohydraulical shifts in the Anchar Lake based on the for the last 6,000 years. For example, during the first phase and the second phase, from 6,000 to almost 4,000 years, and from 4,000 to 1,600 KLERBB, we see that there has been warm and wetter conditions with gradual strengthening of the winter westerlies. As a result of this, there was higher and deeper lake levels. But during the second phase, there was a decline in the precipitation, as a result of which there was relatively shallower lake conditions. During the third phase, that's between 1,600 to 500 KLERBB, we see that there has been some intermittent changes in the precipitation and dry and wet phases in this sedimentary sequence. But during the last 500 years, we see there has been a significant reduction in the precipitation and enhancement in the human influences. This has led to severe eutrophication of the Anchar Lake, particularly towards the margin. Next, we collected a sediment core from the Wooler Lake, which is again located in the Northwest Himalayan region. Here, the idea was to see or observe the short-term controls on weathering behavior and hydroclimate variability in Northwest Himalayas. So, what we did here is we analyzed the sediment core for the strontium isotopic ratios and compared it with the regional hydroclimatic conditions. So our strontium isotope suggested that there has been two well-resolved periods of dominating chemical and the physical weathering in the Northwest Himalayas. For example, during the early phase of from 4 to around 2 KLBP, we see that there is a significant decrease in the strontium isotope ratio, suggesting there has been enhancement in the chemical weathering, because during this phase there would be incongruent weathering of the sediments that would reduce the strontium isotope in the lake base in the lake sediments. But during the last 2 kbp, we see that there has been increase in the or enhancement in the strontium isotope values, which which point us towards the whole rock composition that was derived as a result of the physical weathering in the North Himalayas. When we compare these records with the published records from the Northwest Himalayas, including the some of the other data such as the insolation, North Atlantic Oscillation, westerly monsoon, and INSO record, we see that there has been the strontium isotope better correlate with the higher and lower solar insolation. Here, if you could see this line, you also correlates well um, with our strontium isotope values at 35° north latitude, and temperature anomaly also, which is the temperature anomaly here. This one is the temperature anomaly, and but suggesting that there has been glacial ice cover control on this liquid weathering style. Moreover, we see that the chemical weathering in Northwest Himalaya is independent of the runoff from the tropical subtropical monsoon dynamics, but largely depends on the warm and wetter conditions in this area. Further, we see that glacial ice cover rather than we observe that glacial ice cover rather than surface runoff seem to have impacted significantly the chemical versus physical mode of weathering in West Himalaya. We also did the seaborne ratio of analysis of the sediment of the same sediment core to reveal the which is a proxy of organic detritus supply in the lake sediments, to see whether there has been any shift in this in the proxy in the organic detritus for the last 4,000 years. You see that the seaborne ratio and the WD dominated precipitation, which is this one, actually shows that this region was dominated by westerly mid-latitude westerly precipitation during the late Holocene time period. Could also see some of the different phases during the last 4,000 years. For example, during the early time period, we had the wetter phase, then we had the drier conditions and extreme drought during the last 1.5 to 0.6 kcal/BP, and during the last 500 years, there has been increase in the westerly precipitation and the seaborne ratio, suggesting this phase was colder and wetter in the Himalayas. Next, we did a regional comparison of the dry and wet phases to see the spatio-temporal distribution of the climatic conditions in the westerly and the Indian summer monsoon dominated lake sites. So what we did is we compared the westerly dominated and the Indian summer monsoon dominated lake sites and see what were the paleoclimatic conditions inferred from these sedimentary records. We see that during the first phase, that is the wetter phase in our sedimentary records, that most of the studies imply wetter condition. That's around 60% of the studies suggest wetter condition during this phase. Then we have this was followed by drier conditions and extreme drought conditions during the last two phases. In the final phase during the little ice age, we see that around 64% of the study sites show that there has been extreme wetter conditions in the Himalayas, but a significant opposite relation can be seen here between the Indian summer monsoon and the WD dominated lake sites for the little ice age period. Is that we see that most of these sites show from Indian summer monsoon dominated precipitation show that there has been dry phases. There was a dry phase during the little ice age in the Indian summer monsoon lake systems. The answer to this question lies here. The little ice age in the Himalayas began around 1400 and ended up in 1900. See, if we look at this time period, the area here which is shaded here, which is actually the little ice age phase, is that During this part, we have the, if we look at the ITCZ migration, um, which is basically the intertropical convergence zone, we see that it is migrated toward the south, and there is also a decrease in the solar insolation at this during this phase. And if we look at the glacial accumulation, glacial ice accumulation, and the temperature anomaly, we said there has been increased ice accumulation suggesting wetter conditions, and decreasing temperatures suggesting colder conditions during this phase. But since these Indian summer monsoon lake sites document relatively cold and dry conditions in this during this phase, is that during the periods of enhanced monsoon, the westerlies move north of the Tibetan Plateau, and the moisture-laden southwest monsoon winds penetrate the arid landscape of the Trans-Himalayas. But during the little ice age period, a reverse has happened, that the monsoon got reduced as a result of this southward migration of the ITCZ, as well as the lesser insolation. As a result of this, the westerlies could move south of the Tibetan Plateau and could penetrate this some of the lake sites in the western Himalayas. But there was a significant decline in the Indian summer monsoon precipitation, and hence those lake sites show typically arid or colder arid type of climate during the little ice age phase. So what are the key take home points from this research is that the instrumental and proxy records from the Himalayan lakes attest for the hydroclimate control on the lake sediments during the late Holocene and Etropic evolution for the modern times. The Himalayan mountain glaciers, that's actually the cryosphere cover, serve as important water feeder for the high altitude lakes, and any sort of glacier recession due to prevailing climatic changes would lead to significant decrease in the lake levels. And finally, in order to uh, update these studies, we need to have high resolution time series studies of the lake or freshwater or sediment discharge to further elucidate the subcentennial variation in the permafrost ice melting that has been taking place due to recent anthropogenic warming. So I thank I thank my research collaborators, without whom this research would not have been possible. Especially special thanks to our mentor, Professor Himachutan from Anna University, Chennai. Thank you.
Great, thanks very much. Aasif, that was a wonderful talk. So what we're going to do now is just take 5 minutes for any brief questions for Aasif before we go on to the next presentation. So if there are any questions, you can put up your hand, you can throw it into the chat, or if you want to turn on your mic and just speak, that also is good. Mina has a question right away.
Hi, thanks. Aasif, thank you so much for this very, very informative and nice talk. It's an amazing set of data as well. I'm wondering now in the taking this climate change parameter that we cannot really stop and it's just happening. After a lake is restored, do you have some special, have you established a special operational framework? Have you established framework how to manage the lake and keep it on what you would call a historical baseline, if it's possible to reach that baseline? Because the parameter of climate change, that will continue. You cannot really influence that one. And ecosystems and environments are then not going to be stable within that. So there will be an ongoing directional and environmental change. So the transformation of the restored system will be either slow, fast, or abrupt, again, going to some other state. What is your opinion about that? Is it possible actually to keep the system restored?
Yes, it is possible. but it depends on what is the amount of eutrophication or what is the amount of change that has occurred within a lake shrine basin. For example, if it is in some of the lakes, if it is like the changes are not that significant, so there are some important parameters by which we can restore that lake, or by having it, by keeping it away from the anthropogenic impacts. But we still see that most of those lakes, they have been eutrophicated, or they have been changed to a certain extent where the reversible reaction is very difficult to occupy. But still we see that there are certain proglacial lakes or the high altitude lakes where we can have those baseline values by keeping them away from the anthropogenic impacts, then having those natural pristine conditions that would allow a lake to be at its background condition.
So thank you. So that would mean that once the system is restored, you would think of accepting the climate change going on, and let it change then just within with a very minimal interaction, right?
Yes,
if I understood it
absolutely.
well, because resisting the
stop
climate change is not
environmental change.
okay.
Yes, absolutely.
Thank you very much.
Thank you.
Great. And then we'll have one quick question from Roxanne.
Yes, thank you, Aasif. I wanted to ask a pretty straightforward question. Looking at some of your paleo data, what I found really, really fascinating was, well, first off, thanks, because it was a really interesting introduction to a part of the world where I don't know the dynamics that well, and clearly it's highly dynamic from a hydroclimate point of view. And we're seeing really, really interesting shifts over time that seem to be making the lake express itself similarly. So what I mean by that is, when you have your historical wetter time, your C/N ratios seem to be responding equally as in your sort of drier moments going on, where you're expressing anthropogenic pressures and some level of eutrophication. So it's almost like you have a historical eutrophication driven by wetter climate, and then a historical eutrophication or a present eutrophication, maybe through human activities and those changes. Can you comment on this? Because I found that really fascinating from the C/N ratios and the flip-flop in your isotopic signatures of the oxygen isotope.
Yes, we have seen this recent anthropogenic impacts, but the problem with this the recent data sets is that you do not have the higher number of the higher number of samples, or you can say the high resolution to predict that anthropogenic impact. We have seen this using some of the surface sediments from the same lake systems, where we could see that there was enhancement in the anthropogenic pressure in terms of the nitrification or the higher algal production within the lake systems. But some of the lake sediment cores do suggest some enhancement in this anthropogenic pressure. As we now expansion ratios, we see that there has been some widespread anthropogenic impact, but we cannot clearly comment on that because those samples are very less, or the resolution is very poor for that time frame.
Thank you.
Great, and thanks again, Aasif. If you don't mind turning your screen off, so then we will keep moving forward, and we have the next presentation from Bhakti. I have another pre-recorded presentation that we will try, and we'll see if the mic works. But Bhakti, if you are here, you may need to be prepared to present if the audio has problems again. So we'll see. Hopefully this.
That's absolutely fine. Yeah, again, I think I don't hear the sound, Kale. Should I try to, Bhakti, what do you say? Do you want to try it? Or should I try to share the presentation?
It's probably best if Bhakti presents herself, because I think it would be the same file type, right?
All right.
Yes, give me a
Okay.
couple of minutes.
Sure. I guess in the meantime, if anyone has any questions for, any more questions for Aasif, or any questions about, for the Early Career Committee about the webinar or anything else that ASLO does, now could be a time while we wait. We apologize for the technical difficulties, but I guess this is one of the problems that we have in a global science community, that, you know, now we rely on these types of technologies to communicate, and there seems to be a movement towards this type of communication as opposed to traveling or any other type of regular presentations. But it's very clear that the technological capacity is not equal across the world. So I guess one of the goals of this type of webinar is trying to bridge that gap, but we're also reliant on that technology, and sometimes it's not work perfectly. Okay. Yeah.
Okay, so is my screen visible?
Yep, I can see and hear you fine. Can everyone else please turn off microphones as she presents, and then the same thing when she is finished, we'll do a brief questions directly for her regarding the presentation, and we can have a brief discussion at the end for everyone. So please go ahead and present, Bhakti.
Okay. Yeah, so here we go. Okay, first of all, I would like to thank Amplifying Voices webinar for this opportunity to present a part of my research work, which I am being carried over for past 1 decade. So today the topic which I'm going to present is bioprospecting of halophilic Archaea from the food grade iodized crystal salt from one of the Salterns of India. I am currently working as an assistant professor at the Department of Microbiology, Goa University. Am I audible?
Yes,
Hello.
I can hear you fine. Yeah.
Okay, okay. Thank you. So my talk will have the following subtopic. The outline is I will be talking about the salt pans of India, the salt production process by taking in water, salt water from the sea, how it is done, what are halophilic microorganisms, what do you mean by bioprospecting of halophilic microorganism, and why it is important for us to do so. Now, before starting with the talk, let me speak a little bit about my country, India. Now, this is the image which I have taken from Google Map data. The Indian Peninsula is separated from the mainland Asia by Himalayas. The country is surrounded by the Bay of Bengal to the east and the Arabian Sea in the west, and the Indian Ocean in the south. India is the land of diversity. And now when I say diversity, okay, there are 28 states and 8 union territories in the country. The total length of the coastline of the mainland India and the two islands, that is Lakshadweep Island as well as the Andaman and Nicobar Island, it makes up to 7,516.6 kilometers. And due to this vast coastline, one of the major occupations of my country is salt production or salt cultivation. India is the third largest producer of salt, with the lead producer being China and USA. Now, among the 28 states of India, the major salt producers I have highlighted either with the red highlighter or blue highlighter. Now, the difference is the red highlighted area cultivates salt. Okay, the states, they take up the seawater from Arabian Sea, whereas the blue highlighted areas, okay, these states take up the seawater from Bay of Bengal. Okay, so among the 28 states of India, the major salt producer states are Gujarat, Rajasthan, Tamil Nadu, Maharashtra, Andhra Pradesh, and Odisha. Goa, can you see this, what is highlighted in the arrow red, okay, is the smallest state of India. And I want to talk more about this state because I belong to this state, and whatever studies I have done is from the salt pans from this place. Okay, so Goa is the smallest state of India, and the traditional salt farming in Goa has been practiced for past 1,500 years. Why so? Because Goa has the excellent climate. Goa's riverine estuaries, easy access to seawater, and favorable climatic condition make salt production an attractive occupation, especially during the summers. Now, this is again an image from Google Maps where I have highlighted certain pens and I have given abbreviation CP, EP, and RP. I will explain in my next slide what this is. This is one of the picture which I had clicked during my PhD times when I was collecting samples for isolation of microorganism. And can you see this is the crystallizer pans of the salt pan, and can you see this heaps of salt? It was precipitated and then using certain tools, it is piled up on the embankments and then pulled out together and sold. Coming to the typical layout of solar saltons of Goa and how salt is being extracted from the seawater, let us see. Now, for production of salt, it is a natural process of evaporation where seawater is taken in. Can you see this gate? Okay, this is known as the sluice gate, okay, through which we take up the seawater and we store it in a pan which is called as reservoir pan. Now, we all know that seawater has salinity of 3 to 3.5%. Okay, so this seawater is retained in the reservoir pan until the salinity reaches up to 5%. Once the salinity reaches to 5%, this seawater is then passed on to another set of pans, which is called as evaporator pan. Here, the water is allowed to further evaporate till the salinity reaches 15%. Now, the first salt to precipitate from the seawater in the process of salt production is calcium carbonate and calcium sulfate. This happens either in the reservoir pan or the evaporator pan. From the evaporator pan, the water, or now it is called as the brine. Since it is concentrated, it is passed on to a smaller pen, which are known as crystallizer pen. And as the name suggests, crystallizer pens, okay, in this pen, the salt crystallizes. Okay, so the brine-rich sodium chloride is precipitated as halite. Okay, and what remains back are the salt of magnesium chloride. Now, this magnesium chloride, most of the people call it a bitter because if you taste it, it is bitter. So this is how the salt production usually is carried out in most part of India. Now, what are halophilic microorganisms and why do we need to study them? As we all know, okay, halo meaning salt and philic is something to love for. So microorganisms are living organisms which we cannot see using our naked eye. So we need specialized equipment such as microscope to visualize them. Now here, if you want to still without microscope wants to observe this organism, you can still do it, but you have to culture them out, okay, in the form of visible colonies. So halophilic organisms are salt-loving microorganisms. Okay, so if we wish to study this organism, okay, and if we wish to isolate them, what we have to provide in the media is high salt concentration. So in this slide, I have shown the media on which most of the non-fastidious organism grows. Okay, that is nutrient agar or nutrient broth. And if you see minutely the composition of this nutrient agar, it is having beef extract, NaCl, and peptone. And if you see the concentration of NaCl, it is 5 grams per liter, that is just 0.5%. Now, the carbon source and nitrogen source is served by beef extract and peptone. Whereas the difference between the mesophilic organism and halophilic organism, the media should have excess amount of salt. As I told you all that halophilic meaning the organisms will not grow without salt. Now, what are the salts which are present over here? Can you see over here? NaCl, it is 250 grams per liter. Magnesium sulfate, it is 20 grams per liter. Potassium chloride is 5 grams per liter. And in total, if you consider this, it will be around 275 grams per liter, which is 27.5%, much higher than what is required for the mesophilic organism to maintain the osmotic balance. This is one of the typical halophilic broth which we routinely use in the laboratory, and it is called as the NTY broth, which stands for NaCl tryptone yeast extract. Now, if you want to isolate true halophiles, you have to go to hypersaline regions. And in my state, the hypersaline regions were the salt pans. So what I did, we went and collected the sediment sample and brine sample from these salt pans. Now over here, you can see a picture of a Petri plate, which is having anti-Y halophilic broth. Okay. And if you see minutely, okay, if you see minutely, there is something which is precipitated in the media. This is nothing but the salt, and this salt is precipitated due to prolonged incubation of the agar plate. Okay, what does that mean? That means that these organisms, they like to grow not only at a very high salt concentration, but they also like to grow at very low water activity. That's why they are called as extremely halophilic organisms. And the specialty of these organisms is they produce bright orange red pigments. I will tell you all what is the significance of these pigments in my later slides. So these are some of the isolates which we isolated during my PhD work. And all these isolates, if you observe keenly, they are having bright orange red pigmentation. Now, since this organism we saw, they like to grow at very high salt concentration and almost at a salt saturation concentration. So the next question which arose was, sorry, can halophilic microorganisms survive in salt crystals, especially the salt crystals which we commercially consume? As we all know that, okay, usually we use salt for pickling, okay, as a preservative when we prepare pickle, or even for some fermented foods, we do make use of salt. But we do this salt, do organisms survive even at such high salt or solute concentration? Okay, for that, what we did was we got commercially iodized salt. And this salt packet, okay, was packed or was commercialized from one of the salterns known as Vedaranyam in the state Tamil Nadu, India. Okay, and it was iodized salt. So we took this salt and we dissolved the salt crystals in 15% NaCl. Okay, the minute we dissolved this in 15% NaCl, around 100 microliter, we plated out and we incubated. After 5 days, there was no growth. After 10 days, there was no growth. But minute colonies started appearing after 15 days. And if we observed after 30 to 45 days, the plate was having fully grown colonies. Though all the colonies appeared bright orange red pigmented, okay, we observed them more minutely using stereosome microscope, and they appeared, all of them had different morphology, colony morphology. So we picked them up based on the differences in the colony morphology and further characterized. Now, normally mesophilic organism, they require 0.5 to 0.8% salt to maintain osmotic balance. How this organism survive at 27.5% salt or almost saturation? Okay, so there are two mechanisms which this organism adopt. The first mechanism is to compensate the high sodium chloride outside. They start accumulating high amount of potassium inside their cell, and this is called as high salt in mechanism. Another mechanism is, and this mechanism is usually seen by in extremely halophilic bacteria, is the low salt in mechanism, which is known as compatible solute accumulation. And what are these compatible solute? These compatible solutes are osmoprotectants, and they are small organic molecules which usually have neutral charge. They are not toxic. They can be either sugars, amino acid, or their derivatives. Okay. And they are called as compatible solutes. Why? Because though the organism starts synthesizing or accumulating this in large quantity inside the cell, they do not affect the functionality of the cell. Okay, so they are well compatible with the cell. That's why they are known as compatible solute. Now here you can see one picture, okay? This picture is of an organism which is called as Halomonas. This Halomonas can grow from 5% to up to saturation salinity, and it is known to accumulate one kind of compatible solute which is known as actoin. And this actoin has been commercially exploited, okay, in dermatitis as ointment for protection against sun or as sunscreen. Okay, now these are halophiles. So what is the difference between halophilic archaea and halophilic bacteria? Are they both same? As we all know, the person or the scientist over here is Carl Richard Woese. Okay, he was the one who proposed the 3 domains of life, and the tech which was 16 SR and using tree of life. This tree of life is having bacteria, okay, archaea and eukarya. Now, archaea were formerly known as archaebacteria. They resemble bacteria, but they are phylogenetically separated from the domains of life, okay, because of their unique nature. Now, phylogenetics, it deals with the deduction of the historical relationship between groups of organisms. It tries to represent how every life form is related to every other life form. Okay, so archaea, if we see, this I have taken from a standard textbook, okay? If you see the major difference, okay, between bacteria and archaea, the difference lies in the cell membrane lipid. Okay, in bacteria, it is ester linked. In archaea, it is ether linked. Another major difference is we all know that bacteria, to identify them, the first thing we do is gram staining, which tells us the quantity of peptidoglycan. In gram positive, it is lots of peptidoglycan. In gram negative, it is less peptidoglycan and there is outer membrane. Okay, whereas in archaea, it does not have peptidoglycan. Some has pseudopeptidoglycan and some has something called as the S layer, okay, which can be combined with polysaccharides, other proteins, and so on. The common features are both have 70S ribosomes. Some may have plasmid. They don't have well-defined nucleus. So these are the basic differences and similarities between bacteria and archaea. Now, some of the archaea, okay, have like most of them have shapes which are at par with bacteria, except few archaea, they have different shapes. I have listed few over here. The first one you can see over here, it is Halochondrium welsby. Halo because it is found in salt-rich environments such as solar saltons. Quadratum because if you see the shape of this archaea, it is square-shaped quadrum. Okay, another archaea which you can see, and this archaea you find in sulfur-rich environments. The shape, if you do normal microscopy, that is gram staining or something like that, you will say that it is coccus. But if you use advanced technique, you will say that no, it is not coccus, but it is having donut-shaped structure, involuted like RBCs. And another archaea over here, it is an anaerobic methanogens, Methanosarcina rumen. It grows like a film, okay, or pleomorphic or shapeless. Can you see how it grows? Okay, now this is the basic difference between bacteria, archaea, halophilic microorganism. Now, the last part of my talk is bioprospecting. Now, bioprospecting meaning exploration of natural resources, okay, for molecules which can be commercially exploited, okay, in any sector, maybe agriculture, aquaculture, bioremediation, cosmetic, pharmaceutical, etc. Okay, now what are the specialty of this halophilic archaea which we isolated from the salt pans of Goa or India? I told you all that they have a very unique characteristics that they produce carotenoid pigment. And how do we know that it is a carotenoid pigment? You extract it using a unique solvent or a mixture of solvent. And if you, using a simple spectrophotometric analysis, if you scan the pigment, you will get this typical spectrum which absorbs at 393, 474 to 6, 500, 535 nanometer, which is the characteristic spectrum of carotenoids. Now, these carotenoids, to identify them, or if you run a simple thin layer silica gel chromatography, you will find that it is a mixture of bacteriorubberin and its derivatives, such as mono N-hydrobacteriorubberin, bi-N-hydrobacteriorubberin, isopentenyl dihydrorhodopsin, etc. Now, why this is important? Okay, carotenoids we can obtain from carrots, pumpkin, this, that. But we all know that cultivating this organism, they naturally see it is wild type strains. It is not genetically modified strains which naturally produces such a high quantity carotenoids. Okay, and these carotenoids can be exploited in pharmaceuticals, cosmetics, and even in some food industry. Another bioprospecting or molecule which is is very, very important, and it is important to study this molecule, is microbial bioplastics. Okay, it is known as polyhydroxyalkanoates. These bioplastics were first reported by Morris when he was studying, okay, he was observing the cells of Bacillus megaterium under the microscope. And to his surprise, he observed some granules other than the endospores. Okay, and these granules were granules of polyhydroxybutyrate. This is one kind of subclass of this polyhydroxyalkanoates. Okay, and if you want to see this polymer, okay, they are accumulated as granules inside the cells. You can see this image. This is called as transmission electron microscopy image, where you cut the cell open and you see these granules inside, and they can occupy almost 90% of the cell's volume. Okay, so this polymer is not only important because it is 100% degradable. It is very, very important because these days it has been used in making bone plates, sutures, okay, because of its biocompatible nature. Now, these organisms, what we did, we wanted to screen whether these organisms, Archaea, are able to produce bioplastics. Okay, there is a very simple technique in which you can screen the organism for the production of bioplastic. So it is called as Nile blue or Nile red staining. In this staining, what the stain does, okay, it is specific for the intracellular lipophilic molecules, including polyhydroxyalkanoate. So if the organism is accumulating this polyhydroxyalkanoate, if you incorporate this dye into the medium or attach, flood the cells, okay, with this dye, what it will do, it will go and bind to the PHA. And if this organism is producing PHA, and if you expose the stained cells to UV, it will start showing fluorescence. So the minute it shows fluorescence, that means the cells are accumulating these granules. Now, what we did, we screened 135 halophilic cultures and we found 25 positive for this polyhydroxy alkanoids, bioplastics. What we did, we further characterized who are these organisms at molecular level, and we found that most of them belong to the genus Halogeometricum, Halopherex, Haloarcula, Halococcus, Haloterigina. Okay, and all these are halophilic archaea. And the best producer of this polymer was Halogeometricum borinquens. The strain names are listed here. Then we wanted to see that whether this organism, how much polymer it can produce. Okay, now, always do you think that the organism will produce this polymer? Normally, it will not produce the polymer. It might have the genes for it. Okay, if we want the organism to produce or accumulate this polymer, we need to provide stress to the organism. And what do you mean by stress to the organism? We have to grow the organism in nutrient limiting condition. That means the media should have minimum amount of nitrogen and phosphorus, but at the same time, it should have excess amount of carbohydrate or carbon source. Sorry. Okay. So we grew the organism in commercial substrate as well as agro-industrial waste. I have not included my results for agro-industrial waste because the time provided for me was just 20 minutes. Okay, so I just included the commercial substrate. So when we used this commercial substrate glucose, it gave polymer, which was quite good quantity, but can you see this polymer is not white in color, it is orange in color, because our organism produced carotenoid. And when we extracted this polymer using chloroform, the pigment got co-extracted with the polymer. What we did, we got rid of the pigment. How? By treating it with sodium hypochlorite or acetone. And when we characterized this polymer using all these techniques, we found that it was polyhydroxybutyrate cohydroxyvalerate. That means it was not a homopolymer, it was a copolymer. And always remember, copolymers are having better elasticity and properties as compared to homopolymers, which are mostly brittle. Another applications of this organism was that they produce enzymes which are functional at extreme conditions, okay, where most of the mesophilic enzymes does not work well. Okay, that's why these enzymes are called as extremozymes. Okay, one of the organism, it produced excellent amylase. Okay, it started degrading starch beautifully. Okay, and when we characterized this organism, we found that it was Halococcus. And when we partially characterized the amylase, we found that it was alpha amylase, okay, which had properties where most of the mesophilic enzyme does not function. Okay, that is 25% NaCl, it was active or in presence of detergents, fluctuating temperatures, at varying pH, it was resistant to solvent, and even functional in presence of metals. The last application what I have put is archaeocins. Archaeocins or bacteriocins, when we say, are peptides. Okay, they are proteins which are synthesized by one organism and that inhibits or kills or competes, okay, with related strains. Okay, and as we know, nowadays most of the organisms are resistant. They are resistant to most of the antibiotic. Screening for novel peptides is the need of the hour. And most of these peptides from Halophilic Archaea are yet to be explored for. So we have screened few organisms and we found that they are producing peptide which are inhibitory to related strains. Further studies are yet to be done on this. Okay, so to summarize, the organisms, that is halophilic organisms, especially Archaea, from these solar salt pans, okay, they are excellent producers of many molecules which are having industrial importance. They are also used in metal bioremediation, okay, in the synthesis of nanoparticles using metals. Enzymology, many studies have been done on various enzymes. I just showed one report of amylase. Our other studies are yet to be published. They can be used in bioremediation and even as a model organism in studying how life survives in space, that is astrobiology. These are some of the references which I have used in the study. Okay, and acknowledgement. I would like to, my research wouldn't be possible if I don't acknowledge my supervisor, that is Professor Judith Braganza, under whom I did my PhD at BITS. My colleagues, okay, who were excellent as a teammates, okay, and I learned a lot, okay, from them. Of course, no further studies are possible if you are not having funding or sponsorship. I would like to thank Government of India for the various fellowships during my research career, especially the University Grant Commission, the major research project on which I was working, the Council of Scientific and Industrial Research for funding me as Senior Research Fellow and as a Research Associate, and also the Department of Biotechnology, DBT, for giving me the travel grant, and the current institute where I am employed, Goa University, for always supporting me. Thank you for your kind patience, and it is open to questions. I hope I was audible.
Yeah, thank you, Bhakti. That was a very good talk. Yeah, you were audible the entire time. We have the first question from Roxanne is in the chat, so I'll read that out to you.
Yes, please.
Yeah, thanks, Bhakti, for this talk about these fascinating organisms. The question is, how close are we to using microbes such as these on the creation of bioplastics at a large scale?
Okay, this is the question which, which many of us are, are still in the pioneer stage for this organism, for Halophilic Archaea. But already there are industries set up by recombinant E. coli. However, the best part of Halophilic Archaea is they produce copolymer. Okay, they produce copolymer even if you do not supplement the precursors for the copolymer in the production process. Whereas in case of most of the bacteria, you have to really invest a lot of money, okay, in providing precursors. So suppose I want polyhydroxybutyrate covalorate, then I have to formulate media in such a way. But these organisms are already having genome which are, acclimatized, or they can produce or synthesize copolymer without having precursors added to the media. This is one thing. Second thing is, at industrial scale, we are yet to, propose this organism. Most of the studies are still in the preliminary stage.
Great. Thank you. We have a question next from Danny.
Yeah, thank you, Bhakti, for this very interesting overview of halophiles. I have a general question about natural products of halophiles, not necessarily from Archaea. I know at the time there were explorations into mycosporine-like amino acids as sunscreens produced by halophiles from solar pans, like the one that you described. Is this something that you are following through?
Microsporins.
Yeah, MAAs, so sunscreen, natural sunscreens. I know cyanobacteria, hypersaline cyanobacteria, halophilic produce those.
Yes.
At least.
Yes. So salt pans, they have extremophiles belonging to all three domains: fungi, cyanobacteria, archaea, and bacteria. Okay. Now, I have not focused on anything other than archaea. So I won't be the right person because I have not studied cyanobacteria. If you ask me something related to archaea, I have only focused on archaea and some of the extremely halophilic bacteria with respect to the salt pans. There are studies on fungi, there are studies on cyanobacteria, but no, yes.
Okay, thank you.
Great. We have another question in the chat from Maricela. The question is, do archaeocines affect bacteria or fungi?
Pardon, I didn't get it.
Oh, let me repeat. The question is, do archaeocines affect bacteria or fungi? It is in the chat too.
Okay. Let me read it. Do archaeocins could affect bacteria or fungi? Yes. Archaeocins, when we say archaeocins or bacteriocins, they are produced by one species, such as Holocene. Holocene is produced by E. coli, and it affects related species of Escherichia. Similarly, archaeocins, if they are produced by halophilic Archaea, they are, they are inhibitory to the related species of Haloarchaea. However, we have tested them, okay, against bacteria, and they are showing some inhibitory effect against even multi-drug. Okay, fungi we haven't tried as of now, so they show inhibitory effect. You can say they can be broad range peptides which inhibit cross domains also. Have
Awesome,
I
thank you.
answered it?
Marcella, has that answered your question? Yes, thanks. Another question in the chat from Brittany. How well annotated are the genomes of these microbes? Is it possible to do flux balance analysis or modeling to predict or optimize the production of the desired products?
It is very much possible to do so. Okay, the studies, though there are few groups of people who are actually working on these organisms. Okay, so it has long way to go. We can probably do the whole genomes of certain organisms and do annotation and fetch out the genes for the desired products. Yes, genome guided bioprospecting we need to do.
Excellent. Great. Bhakti, I'll ask you to turn off your screen share. And we've gone a bit over time already, but if anyone is still available to stay on, if we want to have a brief discussion, any other questions for either speakers, questions for us on the committee? There are some of us still on the call. We see some people leaving, so I'm going to share my screen again for a reminder for everyone that the next of the Amplifying Voices webinar will be at the end of January 27th. At the same time, we're going to be hearing again from Justina from Sweden about microbial communities on the seafloor. But if you are an early career researcher from an historically excluded group and you are interested in giving your presenting your science to this audience, please reach out to the committee and we'd be happy to hear from you and organize, hopefully, to get your talk in this slot. And if not, we will have more going on through 2022. Are there any final comments or questions for either speaker or for the group or for the committee? Don't. Richard, and yeah, I see Richard has his hands up, and then Brittany has her hand up after.
Hello.
Hello.
Thank you for the nice presentation. I quite learned a lot within the presentation. However, I have one question. Through the presentation, you showed that the halophilic microorganisms have been isolated on a medium of 25%. This 25%, yes, this is like 250 grams per liter.
Yes.
To my understanding, this is very, very high compared to water, I mean, marine water. The concentration is very, very high compared to that. My question is, why do you need such high concentration to do this isolation? And the microorganisms that are isolated, are you sure that they are not due to some contamination within the processing of this salt preparation? Yeah, that's my question.
Okay, Richard, thank you for your question. Okay, the first question what you have asked is why such a high concentration? So during my PhD, our objective was to focus on only true halophiles. When we say true halophiles are the organisms which will not grow if you do not provide salt for them. Okay, so yes, so 25% salt was the optimum what we used. But when we checked the optimum concentration for this isolate, once we isolated, it was not less than 15%. Okay, so that means the organisms which we have isolated at 25% salt, okay, they were unable to grow below 15% salt. So they were true halophiles. So our first objective was obtained. Okay. Secondly, we could have used 15% salt also. Okay. And we could have, but what used to happen, I had one slide in which the media composition was kept same except for the salt. We isolated using 17% salt and we isolated using 25% salt. The carbon, nitrogen and other sources, okay, in the media were retained same. What we found was halophilic bacteria, the non-pigmented ones, predominated in the media which had less % of salt. So once this organism, bacteria, they grow fast. Once this organism predominate, even if your sample had halophilic archaea, this organism would have masked them. Okay, so there were many things. Now, the second question what you asked was whether it is a contamination or not. No way the contamination will be so uniform. Okay, I have shown you the plates, even this, it was done in, multiple times. So contamination is something which, which is, which you can easily make out. We used to get contamination of fungus, halophilic fungus, but this red pigmented archaea, okay, they were unique.
Yes, if you could allow, I think that I have something else to add. During the questions that were asked, someone, I remember, asked if this bacteria can, the bacteria producing some plastics can be used industrially to produce these plastics. But for me, because you're talking about some product is industry, everyone uses salt. My question here is, did you somehow look into some bacteria with medical issue? I mean, some bacteria that can somehow harm people that could use or people using the salt?
Oh, yes, it's a nice question, Richard. Yes, I forgot to mention that most of this, initially, if we see any of the standard textbook, we used to say that the normal flora of our body is having, most of the time, it had only bacteria, unless the next generation sequencing came into picture. Now, if you see our gut microbiota, okay, because the salt which we consume, maybe because of that, these organisms, extremophilic organism, they predominate in salt also, and even they are reported in some of our intestinal linings. You can check the current reports. So they are not pathogenic. Yes.
Thank you.
And if yes.
Great. I think that this is probably a good place to end off. There's not many of us left in the call. I want to thank both Bhakti and Aasif for giving their presentations and dealing with the technical difficulties in the pre-record, and then also having to deal with the major changes in time zone. It must be quite late for you guys now. So we thank you greatly, and we very much appreciate you coming out and give take.
Thank you, Kate. And thank you. Yes.
Yeah. Great. So yeah.